Hello, everybody, and welcome back to biosc140 Human Physiology. This is the Membrane Dynamics Section 1, Part 4 video. This is material for lecture exam 1. So just a reminder, I did not make a video lecture on cell anatomy. It should be review from a cellular biology course, from an intro to biology course. There will be one or two questions on the exam on basic cellular biology, like what does a mitochondria do? And also on the first checkup, there will be a question or two. You're responsible for this material. So we're talking about membranes in this class, in this lecture. I want to make a distinction before we really get going. When a person says a membrane, there are a couple possibilities of what they could be talking about. So a membrane can refer to different tissues. It can refer to a tissue or a cell boundary. When someone says a membrane tissue, it refers to a tissue that lines a cavity or a sac like a pericardial membrane. So right here we have the heart. Cardio is going to be the heart. So this is a membrane that goes around the heart. It's a sac, a membrane, a tissue, a boundary that goes around the heart. So a tissue membrane that goes around the heart. When talking about cells, it refers to the cell boundary. So a cell plasma membrane, which is a lipid protein barrier surrounding individual cells. So it's a barrier made up of lipids and proteins that surrounds a cell, and it separates outside from inside. Anything inside this lipid protein membrane is inside the cell. Anything outside this lipid protein barrier is outside the cell. So make sure that doesn't get you mixed up. Now, what is the purpose of a cell membrane? What functions does a cell membrane perform? Well, cell membranes perform a number of functions. The first one is the one that I just described. It is physical isolation. Physical isolation. It separates outside from inside. So, I've asked this question a few times so far in this class. How many chemical reactions take place within your body? The answer is a lot. There's a lot of different chemical reactions that take place within your body. And those chemical reactions need very specific environments in order to take place. And one of the ways that your body creates these environments is through compartmentalization. So there's the cellular compartment where there's a certain environment within the cell which allows the necessary chemical reactions to take place within that cell. There are actually membrane-bound organelles. So like the nucleus has a membrane. Mitochondria have membranes. And those membranes allow for compartmentalization. They allow for there to be a specific environment within the nucleus, a specific environment within the mitochondria that's unique, that allows for those unique chemical reactions to take place. So physical isolation, outside from inside, unique environment inside, unique environment outside. Regulation of exchange with the environment. Regulation of exchange with the environment. So you want to have a very specific environment within your cell, like I was just talking about. To do that, you need to be able to regulate what comes into the cell. If you just allow anything into your cell, then how are you going to create a specific environment? So regulation of exchange with the environment. That's things going in and things going out. Communication between cells and the environment. So this is something that I've mentioned in this class before. How many cells do you have in your body? The answer is a lot. There's a lot of cells in your body. And it's very important that those cells function as a team. They function together. They work as a team. I'm going to make an analogy. Think about a team of soccer players that are 5 years old. Think about how they play soccer. They all 11 kids just run after the ball. There's no teamwork at all. The fastest kid is the best kid because he gets to the ball first and actually kicks it. Not a lot of teamwork. Not very good at soccer. Compare that. Maybe your kid's the best 5-year-old soccer player ever. I don't mean to insult your kid, but most 5-year-old soccer players are not very good. Compare that with a high school or college or professional soccer team, and there's a ton of communication, a ton of teamwork between each person, and it allows for the soccer team to be far more efficient. So you need communication in order for your cells to be good teammates with each other. Structural support. So it gives some shape. It gives some shape to the cells. So you've all taken anatomy. Think back to the histology that you learned in anatomy class. Cells have certain shapes because form and function go hand in hand. Why are some cells cuboidal? Why are some cells squamous? Why are some cells, and so on and so on? Structural support, the membrane helps maintain those shapes. It gives some structure. Separation of charge. So we're going to talk a lot about this when we talk about nerve cells. Separation of charge. What does this mean? This means either more positive ions outside than inside, more negative ions inside, so that there's a charge difference across the membrane. Charge difference. So more positive outside, more negative inside, or more positive inside, more negative outside. Separation of charge. Now, what is the cell membrane composed of? Well, the cell membrane is composed of, these things down here. Now, please take special note of these down here because almost guaranteed there will be at least one question about it. And I think a lot of students overlook their slides and it's found on the bottom. So lipids. There are lipids within your cell membrane. So most of our body is liquid. Like water is the main thing that makes up our body. And lipids are really great for making barriers because lipids are hydrophobic. Lipids repel water. Water cannot easily move through a lipid wall. And so lipids are great because they really separate outside from inside in our watery cells. You know, they repel water, and so they're able to repel water on both sides and really form that barrier. Phospholipids are probably the main component of our cell membrane. So right here is a visualized, an artist's rendition of a cell membrane. The phospholipids are going to be these molecules that have the circular aspect with the two tails moving into the middle of the membrane, the circular aspect with the tails moving into the inside. Our cell membranes are actually phospholipid bilayers. So bilayers mean two layers. So we're going to have one layer of phospholipids on the inside, one layer of phospholipids on the outside. We're going to have a few slides on that coming up. And then cholesterol. Cholesterol is another lipid that's a part of the cell membrane. Cholesterols get embedded within the cell membrane, and they can affect kind of the structure of the cell membranes. They give some rigidity, some rigidity to the cells. Proteins. So proteins are very important for the cell membrane also. We're talking a lot about different proteins in the cell membrane, different classes of them, different types. So we have integral and peripheral. So these terms are not really going to demonstrate, we don't really talk too much about the function of the proteins, but more about where they are. So peripheral are going to be on the periphery. Up here, the purple things are the proteins. Peripheral proteins are going to be these proteins that are on the periphery, that are outside, or this section of protein, just this bottom part, just this bottom part. Integral proteins are going to be integrated into the cell membrane. They're going to be integrated into the cell membrane. They're going to be a part of the cell membrane. They might go all the way through the cell membrane, like this one right here or this one right here, or they might just be embedded about halfway into it. That's fine. Integral proteins are either going to be all the way through, or they can even just be into one layer, move into one layer. And then we have carbohydrates. So carbohydrates, let's break these words down. So glyco is going to refer to carbohydrates. Lipid is going to refer to, you guessed it, lipid. So this is a molecule where you have a sugar attached to a lipid. Well, let's find an example out here. Let's find an example. So right here, you see this phospholipid right here? It's got green sugars attached to it. It's a phospholipid right here. It's a phospholipid with a sugar attached to it. So it is a glycolipid. Glycolipid. Sugars attached to lipids. And then we have glycoprotein. So glyco, sugar, protein is, you guessed it again, protein. So you can see over here, you have this carbohydrate, this chain of sugars attached to a protein. So you get a glycoprotein. Glycoproteins are pretty important with communication between the cell and its environment. They do some signaling, and they also provide some structure. So we're going to talk a lot about channels in this lecture, a lot about channels. And so channels, protein channels, one second. Protein channels can be gated or non-gated. Let's see, what does that mean? Well, what does a gate do? A gate prevents things from moving through. If there's a doorway with no gate, with no door, things can just go in and out. Non-gated channels are doorways into and out of the cell that are always open. So non-gated channels are doorways into or out of the cell that are always open, that things can always move in and out. We're going to talk about potassium leak channels when we talk about nerves for test 2. And another common one that you might have heard about is an aquaporin. Aquaporins don't have gates, so the water can just move in and out. Other types of gates, or let me take a step back. Other types of channels have gates. Other types of channels have gates. So a gate is like a doorway. You've got this channel that goes in, that allows things to go into or out of the cell, but you've got a barrier, and that barrier can open or close, like a gate or like a door. Most of these gates are going to have one gate or one door. Some are going to be double gated. If anybody's lived in a place that's really cold, like the Midwest, a lot of buildings will have like a doorway and then a coat room and then another doorway to help keep the cold out. It's kind of like that, like a double gate. We'll talk about double gated channels when we talk about how nerves send signals. So single gated or double gated channels. Now, what's going to open or what's going to close these gates? Well, a lot of different stimuli can open or close these gates. Chemicals can open or close these gates. So a signal molecule binds to or just a certain molecule binds to the channel and it opens up the gate. So it could be a chemical. Chemical would be something binding to the channel and it opens up the gate. Voltage. So this is what I was talking about earlier, where there could be more positive charges outside, more negative charges inside. The amount of voltage difference from the inside to the outside of the cell can affect the opening and closing of channels. Mechanical. So these would be in like the your receptors that sense touch. Receptors that sense touch. So mechanical stimuli can open up channels. Light. Where might we find channels that are opened or closed by a light stimulus? Your eyes. So some channels are open or closed by light. Some channels open or close based on time, and some open or close based on temperature, temperature receptors. So body fluid compartment. So I feel like a lot of students kind of brush this slide to the side, but this actually has a lot of clinical application. It's important for doctors to know how much liquid, how much fluid are in different compartments within a human. So about two-thirds of all of a water within a human is intracellular. So ICF, intracellular fluid, about two-thirds is intracellular. About one-third is extracellular. About one-third is extracellular. Within the extracellular, so within this one-third of the total, you can kind of break it down some. And right here, let's see, let me get my numbers. All right. So intracellular fluid is about two-thirds. So intracellular fluid is about two-thirds. Extracellular fluid would then be about one-third. And of that one-third, about 10.5 liters is interstitial fluid. So interstitial fluid is fluid that is around cells. So interstitial fluid is fluid that's not in cells. It's like the fluid that's on the outside of cells and like is around the cells. About 3 liters of our fluid is found in plasma. So plasma is the liquid portion of our blood. So our blood, not including the white blood cells and the red blood cells, the liquid portion of our blood. And transcellular fluid is usually about 0 to 1 liters. Transcellular fluid is cerebral spinal fluid, so CSF, cerebral spinal fluid. It's intraocular fluid, so fluid within our eyes, and also synovial fluid. Synovial fluid is fluid within our joints, within certain types of joints. So we have total body water, about 42 liters. About 60% of our total body weight is water. About two-thirds of that water is intracellular. About one-third is extracellular. Of that extracellular fluid, most is interstitial fluid, so fluid outside of our cells, kind of around our cells, that bathes our cells. About 3 liters is plasma, so the liquid portion of our blood. And a little bit, the smallest portion, about 1 liter or less, is transcellular fluid, which is your cerebral spinal fluid, it's your intraocular fluid, it's your synovial fluid within your joints. So cell membrane is selectively permeable. Cell membrane is selectively permeable. So over here, we have our phospholipid bilayer, and there's a few things I want to point out about this phospholipid bilayer because it's its structure is very important. So we have phospho and lipid, and we have bilayer. So bilayer means 2 layers. Most of our body is made up of water. We need the membranes of our cells to interact, to play nice with the water that is, that makes up most of our body. Each layer within our membrane, within our bilayer, is going to have a hydrophilic polar head. So it's going to have a portion of the molecule that is hydrophilic, that is water-loving. Hydrophilic means water-loving. And that's the portion that's going to interact with our aqueous outside environment. It's then going to have a nonpolar lipid portion, a fatty acid tail. And that fatty acid tail is nonpolar and hydrophobic. It's going to repel water. We're going to have that hydrophobic tail matched with the second layer, the second phospholipid layer, where its fatty acid, its hydrophobic tails are on the inside also. This bilayer, this nonpolar bilayer, is what's going to keep water out, what's going to separate, what's going to create the barrier between the outside and the inside. And then that second layer is also going to have a polar hydrophilic head to interact with the aqueous environment within the cell. Membrane lipids are amphipathic. So amphipathic is a term that's going to come up a lot in this class. Amphipathic molecules have polar and nonpolar components. Amphipathic molecules have polar and nonpolar components. So permeability. Permeable means a substance can freely move through the membrane. Impermeable, substance that cannot freely move through the membrane. This membrane over here, this phospholipid bilayer, it's going to keep most things out of the cell. It's going to prevent most things from moving through this bilayer and into the cell. Those things that cannot move through are impermeable. The larger a molecule is, the less likely it is to move through the membrane. The more lipophobic or hydrophilic something is, so the more polar or charged it is, the less likely it is to move through the membrane. Sodium ions, they're charged. They are not likely to move through the membrane. Same with potassium, chlorine, and calcium. Glucose is pretty large and it's polar. It's very unlikely to move through the membrane. Substances that are permeable that can move through it tend to be small lipophilic. So gases, O2, CO2, glycerol, and water actually can. It's tiny. It's polar, but it's tiny enough that it can move through. What can move through the membrane? It's influenced by size. The smaller something is, the more likely it is to move through this phospholipid bilayer. Particle solubility, the more hydrophobic it is, the more nonpolar it is, the more it can move through. Particle concentration, the more molecules that are around, the higher the likelihood it is to move through. It's like, how many times do you roll the dice? You know, if there's more particles around, there's a higher chance that one of those particles is going to find a way through. Membrane contents. So we saw in the previous slide that there's different proteins, cholesterol, different things different cells will have different chemical makeups of their cell membrane. And that variation can allow for variation in permeability. All right, and right there, we're going to pause part 1 of this lecture. We'll be back with part 2 in the next video.